So I think we'll get going. So welcome everybody to this webinar with RACI and with the Pharmaceutical Group. So we're lucky to have Andrew Watson here today to talk about high-tech product plant construction. Andrew, as you can see on the screen, is the director, the Centre of Biopharmaceutical Excellence for Asia Pacific. Just a little bit of background on Andrew. I won't dwell too much. We'll get straight into it. So Andrew has nearly 30 years of experience in the design, construction, commissioning, validation, and operation of a wide range of high-tech facilities, includes pharmaceutical manufacturing, high containment, clean rooms, hospitals, pharmacies, specialist research centres. So plenty of experience across those years. Past president of ISPE, or ISPE, I should say, in Australia. And also, I was just chatting to Andrew before we came online about that he's an independent chair of ME60, which is part of Standards Australia, like a committee, I think, for standards and clean room standards, and also a member for some of the ISO standards. And I think he'll touch in more detail on those as we go through. Just give me a sec to make sure we go to the next slide. So all I wanted to do quickly, the last thing from me was that we are using the webinar today through Teams. As an attendee, your microphones and videos will be disabled, so you won't turn those on. If you have any questions, please ask them as we go through the presentation and post them in the chat area in Teams. As you can see, and sorry, the session will be recorded, so it'll be available at a later date. And Andrew has said as well that the slides will be available. So once we've got all that sorted, they'll be available at a later date. So with the chat functionality, you'll see it on the top here. You'll have the little chat icon. If it's got a red dot, it means it's an unread message. And I will curate those questions and ask those questions as we go through. So that's it really from me. So I'm going to stop sharing my screen. So Andrew, would you like to share your screen and we'll get into it?
Okay.
Hopefully you can flick a slide forwards like I couldn't a minute ago. So
right. Okay. You can see, 1 second. I'll just try that again. So you can see that now.
That's on slideshow. That's wonderful. That looks great.
Excellent. Okay. So thanks, Scott. Thanks for the opportunity to talk. I have got around about 40 slides, of which probably about 10 or so I'll be able to whip through fairly quickly and put a bit more detail into the rest of them. It's a bit of a difficult sort of project. This is a huge topic. I've done a lot of stuff in my career and there's a whole lot that I could talk about. So narrowing it down to just this sort of 40-minute presentation and hopefully a bit of question and answer time within that has been a bit of a challenge, but we will see what we can do. I'm going to give you a very, very brief introduction into life science manufacturing in Australia, just where the market's at and a few bits and pieces of that. You may well have better information on this than I have, but I thought it was an interesting place to start. I'm going to talk about facility design, how projects are delivered. I'm going to talk about clean rooms. Clean rooms is probably my main specialty, and it's an area that is very poorly understood. And if you're doing anything in life science manufacturing, whether it's a medical device or a tablet or a capsule or a injectable, it is an absolute key part of the whole process. Talk a bit about construction and commissioning and some tips of the trade that I've picked up along the way on how to handle that. Talking a bit about outsourcing, so what else you can actually get other people to do rather than you doing everything yourself. Talk a bit about where some innovation in the space is, more on project delivery and project project location and those sorts of things, and just touch a little bit on the thing that is usually the first question that anybody ever asks me is, how much is it going to cost? So very quickly, I thought I'd just give you a bit of an idea of where I've come from, so you sort of see where I am. There are some things that you can filter through about whether I may have, particular insight or whether I may have a particular bias or any of those sorts of things. So nearly 30 years ago, I started a little company called IDT in Melbourne. working just as IDT really started to grow and put on facilities. So I was involved in the setting up of all of those active pharmaceutical ingredient facilities, which included a lot of cytotoxic drugs. I did a brief stint in Ireland working at for Merck Sharp and Dome site under project management group. Then came back to IDT, then went to work in construction, which was more on the design and construction of high-tech facilities, which was biocontainment, clean rooms, laboratories, those sorts of things. A couple of years at Pharmout before I went out on my own and with Steve Williams and several other people, Maurice Parlane you might be aware of, started a company called CBE Proprietary Limited, which was essentially a consultancy. And from that, after watching everything go to pot during the pandemic, particularly around the needs for third-party solutions, fill and finish solutions, testing solutions, we set up a company called CBE Pure Solutions, which we are in the process of getting underway at the moment. As Scott mentioned, I was a member of ISPE for many, many years, including a 2-year stint as president for Australia Pacific, chair of the ME60, which is also involved with TC209, which is the ISO 14644 suite of standards committee. And also, I'm also, because I just can't stop volunteering for things or not saying no when people ask me, I'm also a part of the Accreditation Advisory Committee for NATA, which is basically all the people that do all of the work testing facilities. I make sure that I do the sort of the final check on their audits and paperwork. So just looking at where Australia stands, particularly from the pharmaceutical industry, I've got about a few graphs which I'm going to quickly whip through. So firstly, this is the total manufactured exports over the last 10 years or so. This is pre-pandemic. And you'll notice that there was quite a large jump around here in 2018-2019 financial year. So that was around about $54 billion of total manufactured exports. So that's not food and beverages and all those sorts of things. It's pure manufacturing. If we break that down, of that total 54 million, pharmaceuticals and medical instruments come in around about 7.2 billion, which is around about 13.4% of that overall volume. And if we look at that, break that down, there's a word here which that is not really used a lot in the industry. In fact, I spoke to 2 industry veterans and they'd never actually heard of the term. So you break that down into pharmaceutical products, medicaments. Now, medicaments is the French word for medicines. So it's essentially what we would normally call pharmaceutical products, and medical instruments. Pharmaceutical products is a lot of the most of the consumables and those sorts of things that go into healthcare. So if we focus on these two, what we call the medicaments or the medicines and the pharmaceutical products here, and this is looking at a trend taken off the internet over the last, and this takes into account the pandemic. So to the end of last year, you can basically see the blue line, which is exported products that we export, and products in the orange line is products which are imported. You can see that exports are relatively flat, yet imports are rapidly growing. And to me, that senses a bit of an opportunity in the fact that we've got the ability to do more onshore and to do more locally. So there was a paper that was put out every year by AusBiotech. So they've looked at the ecosystem in life sciences, which takes into account both manufacturers, consultants, vendors, suppliers, such as testing companies and all those sorts of things. So there was 2,654 organisations employing over 250,000 people. Nearly 3/4 of those resided in New South Wales and Victoria. And in terms of market capitalisation of these companies on the ASX, it's sitting at around about 233 billion, and that's increased by 63 billion since pre-pandemic. And that basically says that over the last 2 years, it's the highest growth in capital raising on record. Another thing which I thought was interesting out of companies that manufacture or that are involved in biotherapeutics, there was almost about probably a 50% or so increase, maybe a little bit more, between 2019 and 2022, so over that pandemic period. If we look at what the actual manufacturing activity in Australia is, this is based on purely on life science manufacturing. You've got over 400 organisations that have a manufacturing licence, so that's actually over 400 plus sites. Now, of that, that includes testing companies, but it also, about almost a fifth of those are Red Cross donor and processing centres. So if you looked at Red Cross as just one entity, well, then we're probably still talking at around about 320. There's over 150 organisations with an APVMA manufacturing licence, so that's products that are veterinary products. And that's also noting that there are quite a few facilities that handle both human and veterinary products. There are hundreds of medical device manufacturers. They're a little more difficult to actually tie down, but there are quite a few of them, as you're probably aware. There are compounding pharmacies, particularly even sterile compounding pharmacies, which are looked after by various state-based authorities. Pretty much most hospitals would have some form of a sterile compounding pharmacy, which would be a very basic clean room and a cabinet to do work in. And then there's multiple organisations that sort of work in the phase 1 clinical trial. Australia is one of the rare, one of 2 or 3, I think, in the world where you don't actually need to have a manufacturing licence to manufacture a phase 1 clinical trial. That's for safety or those sorts of things. And there's plenty of companies on the fringe. So in Australia, if you don't make a therapeutic claim, as you don't say this product will cure this or will abate this or treat this, you actually don't need a therapeutic manufacturing license. I was at a conference in Singapore last week, and I was talking to a guy who was based, does a lot of CDMO work in Indonesia, and was interested in what was going on in Australia, and sort of asked me the question about, what would compel me to go and work in, to go and manufacture products in Australia. And so we had a bit of a discussion, and this is sort of the things that I sort of put forward. But I qualify each of these reasons to as in with a qualified reason not to. And this is not necessarily my opinion. That my opinion is going to come in next. But this is sort of the general sort of what I would see as the general sort of concept and thought process through this. So we've got a very, very experienced and highly trained workforce, although we don't have a lot of them. We've got the ability to do a lot with a very limited workforce, although labour is expensive. We've got a massive amount of land. If you go to places in Asia, parts of Europe, you'll find that the actual finding the space to do a greenfield site is very difficult, and everything is compromised by land. But obviously, as in Australia, land is expensive and construction costs are fairly exorbitant. We have an extremely well-regarded regulator. The TGA has got an incredible reputation, particularly in Asia. I'm terribly sorry about this. And there's also it's on the basis that it's highly well regarded. It's also because it has the reputation of being a high entry point to get into. So there is a place I used to stay at in Hong Kong, and there was actually a TGA shop, which was basically a chemist which exclusively sold products that had a TGA license. And most of their custom was people coming from the mainland China. This is obviously pre-pandemic and the political situation that's on over there. But that gives you a bit of an understanding about where the TGA is regarded internationally. We were talking about clinical trials. So one of the things that's is unique about Australia is we have a very genetically diverse population. However, the population is small overall. So great place to do phase 1, phase 2, maybe small scale phase 3 clinical trials. Not so good for the larger ones. It needs to be more international. We have world-renowned medical discoveries. However, we have a poor record in translating that to local manufacture. And we are a wealthy country, but we have a very poor entrepreneurial and philanthropic record. Now, if I break that down into what I think are the key issues in terms of manufacturing in Australia, I think the highly trained and experienced workforce and the ability to do a lot with limited workforce is a big deal. And I think if you look at the actual physical numbers of products that have been put out, been manufactured in places like the Pfizer, Western Australia, AstraZeneca and GSK, notwithstanding the issues around the sort of the global toing and froing of where to manufacture, all of those 3 facilities were easily able to compete in China on price, and would be constantly bidding against Chinese factories and being able to beat them on price and be able to meet the demand for volume. That's doesn't, you know, you've got to still got to be part of the sort of the corporate culture or the corporate strategy in the long term, which is why we've lost GSK in Melbourne and we've lost Pfizer in Perth, although Pfizer in Perth has been rebranded at the moment, and why AstraZeneca came so perilously close to going broke over the or to going losing all their products for a period of time in Sydney based on issues around China and China supply. But the bottom line was that they were capable of being an extremely profitable organisation doing that sort of work. Land and construction costs are a problem, and the capital outlay on that is difficult. Spoken about the TGA, genetically diverse population. I think there's opportunities there. Our record in translating to local manufacture is a problem. I think when you have these medical discoveries announced on TV and they say, "Oh, treatment won't be around, you're available for sort of 4 to 5 years," in actual fact, because these products go to clinical trials overseas, we won't see them if they make it through the clinical process at all. We won't see them for maybe 10, 15 years. That's one of the downsides of not translating the manufacture to local. And the poor entrepreneurial and philanthropic culture. There's a lot of money for big stuff and for apartments and all those sorts of things, but the innovative side and things like that, there's a lot of reliance on government grants and a difficult culture in terms of entrepreneurs and local investors. Regulations and guidelines, these are central tenets to what we need to deal with if we're manufacturing locally. So obviously we've got the key guidelines is the PICS Guide to Good Manufacturing Practice for medicinal products. We're currently at version 15, although version 16 has been released internationally. We need a tweak of legislation. It used to be a major change, but now it's just a tweak of legislation to update a version. There's a new Annex 1 of manufacture of sterile medicinal products, which we're still waiting on a version from the TGA when that will be adopted. It is a big deal. It's a document that has gone from 16 pages to 58 pages. It was adopted by the EMA formally as of the 25th of August this year. And it has some fairly significant implications to local manufacture, particularly in the sterile and also the non-sterile. There's a bit of scope creep in this document. Medical devices, broad range of categories of goods. There's ISO 13485 and all the requirements around conformity acceptances and stuff like that. Medical devices is not really my area. I understand what they need from a facility point of view, but I tend to leave other people to deal with all of the regulations around that. APVMA, so the veterinary products. So we have our own special code, which is essentially a very old version of PICS, but it seems to be sort of doing the job at the moment. And one that not everybody would be aware of is there's a range of guidelines on the operation of facilities that handle genetically modified organisms, and that's held under the Office of the Gene Technology Registrar, or the OGTR. So to talk about the design of life science facilities, key things that we need to understand is that life science facilities need to be secure. They need to be under strict controls. They need to be zoned for particular activities to occur in particular areas. They need to be laid out in a logical manner. They need to be safe and efficient. Efficient is a big thing that a lot of designers don't take into account. There is no point having a facility if it's not going to do what it needs to do, which it is in part make money for the people operating it. But the fundamental way that we look at these facilities is work around the flow paths of materials, personnel, and waste. The key issue is that the movement of one material or waste material or person should not adversely affect the other. So you need to take into account the issues around contamination and cross-contamination. So cross-contamination is when you've got two different products and you have a contamination of one with the other. Contamination is just general contamination on a wider basis. Mix-ups. There's plenty of evidence of catastrophes that have occurred in poorly planned, poorly laid out, and poorly managed facilities with materials getting mixed up, and general breaches of health and safety. So one of the other key things that sort of takes into account all of this, what I've just spoken about on this slide is clause 3.5 in the part 1 of the PICS document. And that's that production, storage and quality control areas should not be used as a right-of-way by personnel who do not work in them. So taking into account everything on this slide gives you a basic idea of how a facility is laid out. So if we're looking at the design or the specific areas for personnel and waste and materials, you need areas for change rooms, you need amenities, toilets, lunchrooms, areas for personal storage, write-up areas, meeting rooms, and offices. And of that, the change rooms, as people enter the manufacturing areas, and within manufacturing, these tend to be clean room areas, which I'm going to talk about in a bit more detail in a minute. For materials, you've got all of these areas here where materials come in and where they go and where they leave. And within that, the sampling areas, so sampling of materials as they come in to make sure that they are appropriate and as per their certificate, their C of A. Dispensing and manufacturing, and the primary stage of fill and finish, where you're dealing with the actual putting the product into its final container. They're areas that need to be handled within a clean room space. You need further space for equipment cleaning, washing, drying, and clean equipment storage, although in a lot of biotech facilities these days, as the materials are single use, so you basically bring them in, you use them, they're all pre-sterilized before use, and when you're finished with them, you basically discard them. The actual requirements for cleaning is significantly reduced. Some facilities will have in-post QC, where some quality control tests can be done within the actual facility so they can get immediate results and respond appropriately. And areas where you're going to put all your cleaning equipment that you'll be using to clean the facility. So that all occurs in clean room areas. And then you've got laboratories, and then all of your services area for your air conditioning, your water system, air compressors, liquid nitrogen, all those sorts of things. Then you've got everything that happens around the facility, the access of materials and personnel, egress of materials, personnel, and in particular waste. Service of equipment that needs to be kept outside, your cooling towers, generators, and those sorts of things. Car parking, where cars come in and go, where trucks have to come in and go. Where you need to store large-scale materials such as solvents or liquid nitrogen, and all of your incoming services. For waste is a particular problem because it causes particulate waste in your clean room environments. It provides an opportunity for microbial growth, or may actually have microbes within it themselves, and it's a thing that will attract vermin and pests into your areas. Hazardous waste, so infectious material, GMOs, that's becoming a particular issue with cell and gene therapy and some of the new more advanced medicinal products. Waste that's toxic, and of course waste that's flammable and corrosive that needs to be handled in a particular way. A very, very key part of the GMP is making sure that you have a place where you put any materials that's returned from your customers, anything that's rejected from the processes within your laboratory, or God forbid, if you happen to have any recalls, you need a specific area to store this material, so it could be removed or retrieved as required, and removed and disposed of in an appropriate manner. Autoclaving of waste in some sterile and biotech processes. And in general, you really need to make sure that all waste doesn't interact with process materials and personnel, as we've discussed. For safety, you've got to look at just storage requirements around AS 1940. There's specific segregation requirements based on the amount and volume. You need to have bunding if you've got high amounts. However, even small containers, so this little cabinet here is a 30-litre capacity, so it would store just a couple of bottles of ethanol or something a bit more exotic for your laboratory. But under the current Australian standard and zoning of hazardous areas, you need a 3-metre diameter from each edge of the door of the cabinet. You can have no electrical equipment, power outlets, door swipe outlets, all of those sorts of things that have the potential to generate a spark. And it's 1.8 meters, I think, off the bottom of the storage or the spillage space. That's really can be extremely constricting, particularly in small facilities where you've just got to have a little bit of isopropyl alcohol for sanitation and those sorts of things. Something that's regularly overlooked. Biocontainment, so AS 2243.3, which is microbiological safety in laboratories, is also applied to manufacturing spaces in the viral vector space, some of cell and gene therapies and other biotech operations. This also includes the waste, so you need to include a biocontainment facility where your waste is stored before it goes through the autoclave or is removed for local incineration. Hydrants, hose drills and fire extinguishers all need to be located in particular areas, all need to have exclusion space around it for access and those sorts of things. Safety showers and eye washes. Safety showers and eye washes are a particular requirement under laboratory safety. However, clean rooms are not laboratories. I cannot push that forward enough. And as a result, eye washes and safety showers have stagnant water that comes off the mains. Someone needs to come in who's probably untrained and doesn't really know how to operate in the clean room, who needs to come and operate this thing and take a water sample. So I say no to these in clean room areas, but from an eye wash point of view, and if you're dealing with, most facilities in Australia are small scale, the eye wash bottles, which have an expiry time on it, they're actually sterile and can sit unobtrusively in a clean room environment, are far more effective. So facility types, if you're looking at starting a new manufacturing facility, these are sort of the 3 options that you can look at, and the pros and cons. So greenfield, which is taking a brand new piece of land, building a base building and then fitting out within it, lets you build what you want, provides you with an investment, so something that you can use as a collateral for loan of equipment and those sorts of things. And you can design it to make sure you've got space to grow. However, there's a time penalty, there's a cost penalty, there's a substantial capital outlay. You've got to deal with the planning departments, which are a whole new story in themselves. You've got to deal with authorities such as the water authorities, electrical authorities, and they are completely laws unto themselves, those organisations. And you've got to try and find a workforce. And typically greenfield spaces are usually on the fringes of cities, so it can be difficult to attract people out to that space. Brownfield is essentially taking a facility and fitting it out internally. It can be very fast and cost effective to get yourself underway, but you've got to deal with neighbours. If it was a warehouse and you're turning into a manufacturing environment, you've got changes of use, so that goes back to the planning department. You've got to make sure you've got sufficient power. Where is your waste and sewerage access located? How do you get access? All of those sorts of things, and can be growth limiting as well if you can't find the right space. Institutional, so this is basically taking space within a university or a institute, research and development institute and things like that. Fantastic way of attracting smart people to work in your organisation. Your investment can be significantly reduced. You don't have to worry about planning or authorities, but again, you've got to work within an R&D facility or other things that are going on within this space. You've got shared services that you've got to deal with, and space that you've got to share, and obviously growth limiting as well. So the attributes of a facility, obviously if you're talking, but this is particularly if you're talking about a brownfield space that you're going into. Obviously needs to be integral, secure, vermin-proof and weatherproof. The floor needs to be flat. The floor needs to be integral as well. And I cannot stress this enough. If it smells bad, you should run a mile. If it smells like a cabbage or something like that, you've probably got an open sewer somewhere, which means the place is going to be full of all sorts of terrible things. And you're always going to have a micro problem. If it smells like mold is almost impossible to get rid of. And that's a starting point of saying, no thanks. You want to have a decent floor to floor or floor to roof space, so you can put air conditioning and service equipment above your facility. You need to be careful where you're located. You don't want dirty industrial areas or areas under active development where you're going to have lots of dust and trucks and all sorts of people coming and going. Spaces that have neighbours that have a high buyer load, such as bakeries and breweries and those sorts of things. And also the issues around proximity to residential areas. As much as they like to see that people are making drugs, they really don't want people making drugs next door to them. Power, you need to make sure you've got, even for the smallest project, you need at least 100 amps. And most facilities these days are supplied as a base load of around about between 50 and 70 amps. Sewer location, you want to make sure that your sewer is in a space where you want it to be. You don't want it to be at the back, or you don't want to be at the front if all of your wet areas are going to be at the back, and vice versa. And you need contractors that have the reputation of attention to detail. They need to be very proactive, and they just, not just doing as the architect tells them, which I'll go into in a bit more detail. And these are just a few red flags. One of my two that at the top are my real bugbears. So clean room with two words instead of one word, and I'm going to give you a bit of a thing to remind you on that in a minute. And people calling their clean rooms laboratories. They are not laboratories. They are places where you manufacture things that will be going into a human or an animal. So what is a clean room? So this is a definition from ISO 14644 Part 1. So I mentioned before about TC 209, which is a organisation or a committee that looks after these and writes and amends and reviews and revises these documents. The ISO 14644 Part 1 standard is one of the top 10 selling ISO standards in the world, we're told. So it is quite widely used. So this is where we talk about a clean room. So clean rooms are defined by the number concentration or the number of airborne particles. So we're not worried about what's on the surface, we're worried about what's floating around in the air, which is designed and constructed and operated in a manner to control the introduction and generation and retention of particles within the room. So it is a specific space where we're not trying to make it perfectly clean, but we are controlling it to a particular level commensurate with what we actually want to do within the facility. So there's a whole lot of other attributes that we will also potentially want to control, such as bugs, like microbes, such as chemical vapors, or smaller particles down to the nanoscale. But particularly in life science clean rooms, we are concerned predominantly with airborne particles and with microbes, which is a separate sort of thing, which we'll talk about a bit later. And for every clean room, the particle concentration is specified. So as a note, a clean room, one word, is a room that's been quantified through ISO 14644 Part 1. So someone has come in and tested it and said, yes, this is an ISO X clean room. A clean room, two words, is a room that's been qualified by somebody. They've looked at it and said, yep, that looks clean. And that person is typically a mother. So clean room, very, very technical term. Clean room, two words, anything can be applied to anything.
Sorry, Andrew, just quickly, you mentioned the laboratory a couple of times. Now, my background's testing. Why are they getting lumped in with laboratories? I don't understand how that would happen, because you seem to say it happens all the time by what you've said, I think.
So the main reason for that is that most contractors will have dealt with laboratories before.
Right. Okay.
So and because they see it as technical, that's just there, and they see that's, you know, places where people walk around in white suits and those sorts of things, they just assume that it's a laboratory. And this is a big issue in Australia, because Australia has a very, very strong biocontainment bent. And there are particular ways that you design and operate biocontainment facilities and fit them out. And biocontainment facility fit outs are done specifically for biocontainment. Clean rooms are fitted out in a very, very different way. And I find a lot of clean rooms fitted out like biocontainment laboratories.
Right.
And that's a problem, because as soon as the language sort of creeps in, then it becomes normalized that, okay, they come out of that idea of what that they're working with a clean room, go back into what they're used to with laboratories, and you have design issues as a result.
Yeah, and to be honest, laboratories that I've worked in sometimes aren't even particularly clean. But anyway, that's a different
matter. Totally.
Thank you.
Okay, so there are two types of clean rooms. And it really comes down to how air is moved through the facility. So fundamentally, we have got one which is unidirectional, which is where you've got a full HEPA, a ceiling full of HEPAs, and basically air comes down like a piston. And it essentially, you've got clean air that displaces or pushes any contamination generated out of the critical zone. You'll find them in some operating theatres. You'll find them in large-scale microelectronic facilities, and in biotech, we generally try to condense them because they're incredibly expensive to design, build, and operate. Find them what we call separative devices, which is like Class 2 biological safety cabinets and clean air devices and those sorts of things. The second type is what we call non-unidirectional. So that means we've got a scattering of HEPAs around or not even necessarily HEPAs, but a scattering of filters and air that basically means that you've got clean air that comes in and mixes with the room air, and it dilutes any contamination as it's generated. Now, there is a massive sort of 4-hour talk that I could go through on that, but I won't, because we're running a bit short of time. But that's just the sort of the two things to be aware of, either unidirectional or non-unidirectional, displacing contamination or diluting contamination. So there are three key sources of contamination. One is the facility, two is the process, and three are people. And if you break that down, the facility is particularly if it's you've got a HEPA filtered supply, that amount is tiny. For the process, for non-sterile processes, we're dealing with tablets and capsules and liquids, cough mixtures and those sorts of things. It's a moderate amount, but again, tiny amounts for sterile. And the main one is people. And that's the huge source, the most major source, and the thing that we're spending most of our time when we design clean rooms is dealing with the contamination shed by people. Contamination size. Basically, we perform using special instruments. We take the air and we actually have the ability to be able to count the number of particles in the space that are greater than or equal to a particular size. So in life sciences, we really look after products, a particle contamination that is 0.5 micron and above, and 5 micron and above. Above that, we're talking more specialist areas, and below that, you're sort of talking about the more high-end lithography and silicon chip manufacture and those sorts of things. A key thing to understand is that microbes and also to another extent viruses don't hang around as sort of single entities. It's pretty much they move around because they're attached to a piece of skin or something, some sort of process that's been dislodged, and they float around attached to those. Now, a key thing to remember about particles is that particles that are 5 micron and greater tend to settle to the floor, whereas the smaller particles will be easily entrained into the airstream and removed. And so one of the key things to remember is that most of your contamination is that you're worried about, because if you're in life science, you're worried about microbes. They are generally on the floor. They move around when people walk around the space, and people tend to kick them up into the air. If you want to get a bit more of an idea, this is a really interesting thing to look at. You'll probably never look at people again in the same way when you see just how much contamination they tend to shed. If anybody's interested, yes, women do tend to be cleaner than men, although particularly clean women are much more cleaner than the clean men, but the ranges are quite phenomenal, and it's really quite an interesting paper to read. So I've spoke about the 14644 suite of standards. So these are, I'm just going to put here for your reference. These are the key ones. There are about, because a couple have been removed, we've got around about 19, I think, documents, and a few more in preparation. Part 1 is the key one that you'll always see referenced. Excuse me. Part 3 is also what we use to test your clean rooms, and we've got some other documents that are a bit more sort of esoteric, but will be coming more into play as you sort of become more aware of how clean rooms operate. So as I said, part 1, so there's an Australian mirror standard. That's why we've got an AS in front of it, and that's what your clean room testers are accredited to operate to. Details how clean rooms are classified and how clean rooms are tested to show that they meet their designated classification. Now, when we classify a clean room, a lot of people just say, oh, this is an ISO 8 clean room, an ISO 5 clean room, or whatever. It actually matters a great deal about what is actually happening in that clean room as to what that classification is. And that's based on what we would call our occupancy state. So there are 3 occupancy states: as built, which is essentially what the actual room is; at rest, which is what contribution the room and the equipment make to the cleanliness of the facility; and operational, which is where it's operating in its regular manner, or sometimes I think under PICS PICS GMP we talk about operating at risk. So it's these 2 things that we use for life science facilities. Now I've got some tables which I'm going to very, very briefly touch on. I've left them in here mainly for your review and if you for something to have and to look up. I've probably cheated ISO out of someone buying a standard, but that's too bad. So this is table 1. This is how we specify the cleanliness or the cleanliness threshold of a clean room. And that comes, we've got our ISO class number running down here on the left, so from 1 to 9. We've got the maximum allowable concentrations for particles equal to and greater than the considered sizes, from 0.1 micron up to 5 micron. And these numbers here are the actual, the numbers themselves of the maximum concentration of particles per cubic meter. This is translated from ISO class into a grade, particularly for the manufacture of sterile goods. So this is what's in the current Annex 1 for both. So I've got both here, the particle concentrations and the equivalent of in ISO. So within grades A, B, and C, there is a at-rest component and an in-operation component. So if you've got a grade C clean room, you will test the clean room twice. Once at rest, in which we expect it to meet an ISO 7 standard, and once in operation, where we expect it to meet an ISO 8 standard. And then attached to that are a range of microbial cleanliness levels, which is all based down here, and as part of a classification of a facility and qualification of the facility, these are the settings that you need to meet. There's some changes within the new Annex 1, and we've split it down into the work that you need to do in classification, so when you're actually classifying the rooms, and then the ongoing monitoring. And the amount of monitoring you do is commensurate with the level of cleanliness, so the cleaner the clean room, the more monitoring you need to do. And then so within the new Annex 1, there's a separate set of tables that shows how you need to monitor to maintain in between your classifying of your clean room, that you're maintaining the required cleanliness level. So if we just quickly just go on what the difference is between grade A, B, C, and D. So grade A is used for the filling of high-risk sterile products. Grade B is the background to grade A. So if you've got a biological safety cabinet or a clean air workstation where you're preparing sterile materials, you'll have a grade B background where the personnel will actually stand. Grade C is for filling of products that are going to be terminally sterilised or in preparation of solutions that are later going to be filled in grade A. We'd use this for the downstream biotech processing for a closed system process. We'd use grade C. And then grade D, preparation of solutions that are going to be filled in grade C or to be used in the upstream process of a biotech. Sometimes people just have grade C in both. And it's also applied to non-sterile production. So and again, this is from the new Annex 1, there's going to be much more of a focus on looking at some of the attributes or requirements of Annex 1 and that flowing into non-sterile production, in particularly around ointments and creams and cough mixtures and the like. Cleanroom HVAC, again, I could probably talk for most of the day on cleanroom HVAC, so I'm just going to give you a really basic top-level overview. We control temperature. We control pressures for the exclusion and containment of contamination. Sometimes we control humidity. Most people do it when they probably don't need to, but it is a component, particularly in life science, particularly sterile operations, you need to be aware of. For pressure control, you fix the supply air, you vary your return and exhaust air. So if you want a positive pressure, you put a little bit more air in than you're taking out. Or if you want a negative pressure or a lower pressure than surrounding areas, you take a little bit more out than you're putting in. But a fundamental thing to remember is that for non-unidirectional clean rooms, you provide sufficient air to maintain a specific clean room class or grade in operation. There's a whole lot of other rules of thumb and stuff like that that people get tied up in terms of air change rates, in terms of the number of airlocks to get into a particular clean room and stuff like that. Fundamentally, that's what you're trying to achieve. Filtration, HEPA filters are ubiquitous, but they're not necessarily essential for some non-sterile grade D clean room areas. You always have your supply at high level. You should always try and have your return at low level. That gives you a much better clean room. You can have an exhaust either up or low, depending on what you're trying to do with it. And you need to make sure you've got sufficient distance between a supply and return, because you're providing this clean air into your clean room to dilute your contamination. You need to make sure it's well distributed. Controls, there's lots and lots of mothballed facilities because the controls have been far too difficult to actually provide a stable facility. So always say keep it stable, keep it simple. And the key thing, a building management system, so that's what actually controls your clean room. It's important, you need to know how it works, but it's not something that's the be all and end all. The be all and end all in pharmaceutical or life science manufacture is your environmental monitoring system. And that's an independent system that lets you know that everything is okay. Again, that's another whole day in itself to go through all of that. Separative devices. So this is something that where you, for sterile and biotech processes, where you do your final fill and finish, or where you need to do a final level of containment, provides a level of separation between the internal and the external. Can either protect staff from the product or product from the staff, and sometimes a combination of the two. This is a list of some of those things: biological safety cabinets, isolators, RABs, those sorts of things. Doesn't include biological safety, class 1 biological safety cabinets, or fume hoods, or powder containment booths. They're typically things where you stand in front. They only provide operator protection. And if you've got a clean product in there, as I saw the other day, if you've got a clean product in there, all the air's passing past you or the personnel who's doing the work and contaminating your product. So this is just a representation of each of these things here with a bit of a simplified flow diagram. So for more the sort of the details of design, there's a whole bunch of things here. I'll leave that for you to have a bit of a look at. I could probably have done another 30 pages of these, but I think these are sort of really simple sort of things just to think about going forward. I've got a few photos here of common problems that I see where I go. Mechanical guys they hate to get up into ceiling spaces. So every chance they'll stick an access hatch in your clean room. If you open that access hatch, you'll need to shut your facility down and do a full re-clean. Also, as it being on the ceiling, and because the rooms are pressurized, as you open and close doors, you will most definitely draw in contaminated air from the ceiling. So these need to be completely sealed. Even though if you've got to cut the silicon to get into them, they need to be completely sealed. So really they should be avoided at all costs if possible. Brass fittings in the clean rooms. Brass loves to corrode under normal cleaning chemicals, so that could be a problem. Whenever you have a TGA or an APVMA inspector, they'll come and they'll look at all the perimeter doors and see if they can see light underneath it. So you've got to make sure those gaps are clean. The tops of doors is a favourite thing of mine. Usually they're just exposed wood because they haven't been painted properly or coated properly. But this was a beautiful stainless steel door which had a wonderful gap at the top where every time it cleaned, moisture was getting in there and basically rotting the cardboard on the inside. And then on the right, again, people not applying silicon in a clean environment, getting contamination in the silicon, which then remains there for the rest of the life of the facility. Some things to expect of from your builder. You want a program, a budget. A clean build protocol is rarely used, but it's a really interesting tool and useful tool for controlling the amount of contamination that's generated as you go through and making sure you've got a clean and easily validatable and qualifiable facility at the end of it. In terms of rarely implemented, a comprehensive manual on the day of handover is also rarely implemented, and it's one of the things that you really need to keep on the backs of people to make sure that's delivered. I'm just going to leave this up here. This is a little bit of an idea of how a facility is installed and commissioned all the way through to validation. A lot of my clients have found it quite useful, so I thought I'd just add it to this document for you to have a look at. Commissioning qualification, just a couple of key things here just to note. Number one, when you're commissioning a facility and you've specified what your temperatures are, your humidities is, what your pressures are, and all those sorts of things, they're not just a single watching a gauge and seeing the number and then saying, yep, tick, got it. It's got to be, you've got to allow a certain period of time, like a 24-hour period or a 7-day period even, to demonstrate that the facility is able to maintain over that duration. And it's the thing that holds up the finishing of a commissioning of a facility. Number one, trying to get the pressures to balance, but number two is actually trying to demonstrate that the facility is actually stable. Probably another thing, particle counts, the at-rest particle counts, so that's without the actual operations of the facility, should be performed by the contractor, whereas under qualification, the in-operation particle counts should be performed by the client, because only you know how the facility is operated. Again, qualification, I could probably give you a week discussion on that, which we won't go through, but a couple of things just to note. If you've got a sterile clean room, that takes a long time to qualify, 3 to 6 months. Water systems, I always work on the basis of 6 to 12 months, although you can sort of get undergoing with water trials and validation trials and stuff like that within this period here. So in terms of some innovations in facility design, so modular, transparent, transportable facilities. There's a whole range of facilities that are available overseas. They're great for getting an international standard, but to be honest, they're very, very expensive. And one of the things about Australian Building Code is your electrical and plumbing and data and those sorts of things need to be installed by a qualified Australian installer. You don't get that if you're getting it done in Ireland or China or the southern United States. There are ways around it, but no one really seems to be able to give me a reasonable idea of how that can be done. Local units, they are expensive, but not as expensive. You probably spend about a 10% sort of premium on having something that's modular and potentially transportable. And just as a thing, this is our facility in Melbourne. The white building you can see here are 4 modules, 10 metres by 3 metres. They're 2 separate facilities owned by 2 separate companies, but the one serves the other, and they weigh about 3.5 tonnes. Each module weighs about 3.5 tonnes and can be picked up and moved relatively efficiently and effectively. Shared and multi-use facilities. Now, this is, I mentioned this before in talking about institutional buildings, and this is a fantastic opportunity for startups and for people going through clinical trials who don't want to invest in a facility maybe overall or maybe just at the start. But there are significant regulatory hurdles. And the key thing is, if you've got 2 facilities that are being shared by 2 different people, you need to dictate who is in control of what particular space. The TGA can't stand that idea that 2 people being under the same control or working under 2 different quality management systems. Something to think about. But I think it's something that where things are heading in the future. Outsourcing, even the largest facilities outsource to a degree. Fill and finish is probably around, if you've got a sterile facility and you want to do fill and finish, the fill and finish equipment is probably going to take up around about 30% of your budget and probably about 50% of your time in regulatory compliance. So a lot of people send that off. Micro testing, chemical testing, bioburden testing, sterility testing, all of those things tend to be outsourced. Production staff, so you particularly if you're starting up, you don't want to have 40 people sitting around waiting for your process to get underway. It's always good to bring in outside people, and there's plenty of companies out there that can do all of that. And then all of the other things, particularly around qualification and validation, API manufacture, those sorts of things. So finally, what should it cost? So if you're talking about a greenfield government project where you've got a massive oversight, architects by the dozen, engineers by the dozen, everybody covering their bases and making sure that everything is as it should be, you can be looking at upwards of 20 to maybe even $25,000 a square metre. If you're building a clean room, it does depend on how you actually and the level of expertise and the way that you're doing it. But I mean, as in a box on ground for sterile biotech, you're probably looking at between 10 and 15,000 a square metre. For non-sterile, for tableting and liquids and those sorts of facilities, around $4,000 to $7,000 a square metre. And that would include your building fabrics, so your walls and your ceilings, your flooring, air conditioning, furniture and hydraulic, and would exclude critical services and equipment. For other costs, fill and finish machines can cost anywhere from $500,000 to $50 million, depending on the size. I have did do a price for an approach to market for an mRNA vaccine over the pandemic that came in around about 50, and then the facility itself was probably about another 100 million on top of that. Didn't go ahead, of course. Water systems, for a small purified water system, you'd expect to pay about 100k. Small WFI anywhere between 500k and $1 million, and then add zeros from that if you get larger. Environmental monitoring system, 50 to 200k for non-sterile. Sterile, where you're looking at having to bring on online particle counters, could get you up around the sort of the 400k for a large system. Pharmaceutical quality systems, not everybody who's starting up has got the ability to put all this together. It can take 6 months, it can take 18 months, it can take 2.5 years, I've seen. And it depends on how you sort of fund it. The best way, I think, is to partially outsource it. So you've got someone that's feeding you details and providing support on a small team that you've got, and you can spend anywhere between 550 and 100,000 there. If you're fully outsourcing it and getting someone to do it and handing you over something at the end, then you can be spending anywhere up to $500,000, depending on the size of the facility. Qualification and validation, always as a rule of thumb for facilities and equipment, allow 10 to 20% of the total cost, and that would include things like outsourcing for getting environmental monitoring work done, testing, and those sorts of things. And finally, quality manager, if you can find one. They are in incredibly short supply, but you would be starting for a small organisation. You'd be looking at 150,000 and then going on from that. So that was a lot of material. Hopefully there's something within this. I'd recommend that you look over the document and you have a bit of a read. All of my contact details are there. So if you want to ask me any questions after this, or you have a bit of a revelation at 3 in the morning or tomorrow in the shower, certainly feel free to give me a call.
Thanks, Andrew. That's great. We have run over slightly. Hopefully, we've only got one question at the moment. I'll see if are you okay if a couple come in to stay on for another maybe 5 minutes or so. Is that all right? Or
I've got the rest of the evening.
No worries. We have some dinner at some point. But there was a question early on about what requirements exist around cosmetics. Sorry, I kind of missed it when you did it earlier, whether you covered that or not, or whether you get involved with cosmetics at all.
So for cosmetics, so a particular cosmetic would be especially cosmetics that have sunscreen. So there are some specific requirements around sunscreens, and you need sort of a clean environment, and there's a whole lot of regulations around that. So you do need a manufacturing license for that.
Yeah.
For some cosmetic, it depends if you're making a claim. If it's a therapeutic claim that you're making, then you'll need a TGA license. But other than that, although a lot of facilities do operate at a low-level clean room or just below a low-level clean room space, you've still got to worry about microbial control and those sorts of things. But the actual requirements are fairly limited.
Yeah, I do remember, like you said, like sunscreens are regulated, not in that many countries, but yeah, as soon as it's say over, I can't remember, SPF 15 or something or whatever.
Something like that, yeah.
Yeah, but yeah, if it's a pure cosmetic, yeah, I'm not saying you're allowed to do what you want, but you know what I mean. Like you just don't want to hurt anybody, do you? You know what I mean?
No,
like,
exactly right. But obviously, cosmetics, there's a whole range of things around that. And the regulations, I mean, again, if you're dealing with cosmetics where you're doing something for a named patient under the exemption 5A, you actually don't even need a TGA license, for better or worse.
Yeah, fair enough. Fair enough. Was there any other questions from the attendees? Anybody furiously typing? I can't see any else coming through at the moment, unfortunately. But if not, we'll knock off in a minute. Or maybe I'll give them 30 seconds, see if someone's got anything come through. Oh, sorry, I did miss one. Sorry. What issues do you typically run into when converting lab space or existing facilities to clean rooms or manufacturing sites? The doors don't shut properly. Maybe I've got a gap underneath the door.
Yeah, or there's look, again, that idea of integrity and security and pest control and all those sorts of things. Look, a classic one is you've got all of your wet areas that if you've got to put in somewhere, say you've got a warehouse that you're converting, and you're going to have all of these wet areas there. Digging up the floor to get drainage across is difficult. If you're setting up at the back of the facility and your sewer is at the front of the facility because you've got to have a fall, you've got to dig down really, really deep, and it's disruptive, and you're generating all sorts of contamination and those sorts of things. From laboratory to clean rooms, one of the biggest issues, and I touched on this, is power. Is generally laboratory, the difference between a clean room and a laboratory of the same footprint is probably around about an extra 30 to 50% power.
Really? Right, okay.
And if you've got to upgrade the power to your facility, it will, the authorities are an absolute law unto themselves. I know of a facility that wanted to upgrade by 100 amps, and they got an initial sort of, it's going to cost you $100,000, and then it took 6 months to get that, to try and get the price confirmed. And when the price came back, it was $6,800. And then it was going to be sort of a 12-week sort of process. I've seen facilities where they've been told it's even just a minor upgrade is going to cost them $2 million because they've got to put a transformer, a kilometre down the road. So it's, anything where you're dealing with an authority is a problem.
Yeah. I've seen it personally on a much smaller scale, just to get them to sign off on the building. You could be in there 2 years sometimes before you even signed it off. It's bizarre, but anyway, let's not go into that. There's a question around quality managers. You're saying they're in short supply. What kind of qualifications are expected, was the question?
Generally, and this is an interesting one, because the TGA, if you're having like a pre-licensing inspection or something like that, or you're having, discussing your prospects with the TGA will ask who your quality manager is, or who you intend to be your quality manager, and they will ask about their qualifications. So generally, most people would expect you to have a microbiological background, be a microbiologist, particularly if it's going to be, or essentially if it's a sterile operation. They'll probably insist on it, and they would expect that you've got between 5 to 7 years of quality experience, preferably 10. But if they don't think the person's got sufficient experience, they will tell you, and you'll need to listen.
Yeah, fair enough. No, that's fair enough. There's a question around what problems occur from having food manufacturer next door. You gave bakeries and breweries as examples.
Yep.
I suppose they've got live cultures everywhere, I'm guessing, is why you said that.
Exactly right. Yeah, but yeast going everywhere and worse. So
other food manufacturer in particular, or did you say?
Yeah, look, there's, I mean, the other obvious thing is how they handle their waste. They're not under the. I mean, they still will have pest control programs, but they might not necessarily. They're not held to the same standard as you would be. So yeah, and of course, depending on what you're doing, they may well have an issue with what you're doing, and may complain and take you to the Administrative Appeals Tribunal and all those sorts of things. So there's all sorts of issues, but from what I've, from my understanding, is it's really that yeast and bioload that you get with those sorts of industries that are a bigger problem.
Cool. By the way, Annette, who asked the question about the quality managers, happy to come out of retirement and earn some big bucks.
Yes,
apparently.
absolutely.
So fair enough.
Well, if she's out of retirement, then she'd probably double that salary if she wants.
Yeah, there you go, Annette. You can have 300 grand a year. Sorry, a clean room question. So clean room standards for non-sterile API manufacture, Class E and F, have been mentioned in discussion papers. How do you see these standards in the context of new Annex 1?
Yeah,
you seen that E and F? Is that a European thing, is it? Or
possibly. The minimum, and this is, so there is discussion. There's in the opening statements of the new Annex 1, they rather ominously state that many of the requirements within this document can be applied to non-sterile, and that was a red flag from the get-go on the fact that this is not necessarily now just specifically for sterile. if you are dealing with non-sterile, particularly if you're dealing with liquid products, I would say grade D/ISO 8 is an absolute minimum.
Mm-hmm.
I mean, essentially ISO 9, which is an order of magnitude lower than ISO 8, is essentially the environment that you're sitting in at the moment. Achieving ISO 8 or grade D is not really that difficult. And if you look at it from a microbial perspective, 200 CFU per cubic meter is pretty dirty. That really is quite horrendous. You wouldn't expect to see that very often at all. So the idea of ENF, I know way back we used to, when we were talking about the actual concentration. So we used to have in Australia we used to say class rather than ISO 8 or anything like that. We used to say class 3 or 3.5 for grade A, class 35, class 350 for grade C, and class 3,500. We then applied a class 7,000, and that was popular in tablet manufacturers and things like that.
Right.
The key thing to remember is that typically for non-sterile, when we say ISO 8, we say ISO 8 at rest. And at rest is a relatively easy, they expect that if you're dealing with a whole lot of powders and capsule dedusters and all those sorts of things, that you're going to have a much higher load while you're in operation. But the basis is if you can bring it back to ISO 8 at rest, then that's a good demonstration that you've got a degree of control.
Because no one's moving around, are they? Or
no, there's no one
people
inside.
or stuff. Yes,
no,
yeah.
that's
Fair
right.
enough. Yeah, it seems interesting. I'm certainly no expert, but D was a relatively, like you say, relatively low bar. E and F, I don't know what you've got there. You're outdoors, by the time you get to F, I don't know. But anyway, we'll work out. But no, that's great. Look, and that's wonderful, Angela. I think we'll leave it there. Thank you again for your time. It's been fantastic. Fantastic thing. And as you said, your stuff's still on the screen there if anyone wants to contact you or CBE. And as we said, the recording will be available. Just got to, edit it a little bit, make sure we've got all our rubbish at the front. And you'll provide the presentation, won't you? We can post on our website, in maybe in a couple of weeks or something like that. So we'll have that already.
Send it through now.
All right. Thank you very much. Thanks again and have a good evening. Enjoy yourself. Thanks everybody for listening. Thank you.
Okay, bye.